Okay, we're going to run through part 4 of Chapter 7's lecture. So, in terms of moving water across the cell membrane, what we need to look at is the concept of osmosis, which is the diffusion of water. So, again, diffusion is moving from high concentration to low. So, osmosis is moving from high concentration of water to low concentration. In terms of living organisms, and when we're talking about the cell membrane, we add into our definition of osmosis the diffusion of water across a semi-permeable membrane. So that's an important difference that we need to consider. So part of that brings us to the definition of tonicity. So you'll hear the term hypertonic, hypotonic, or isotonic. And the general idea that we're talking about is the ability of a solution to cause a cell to gain or lose water. That is the definition of tonicity. And it depends on the membrane and how permeable it is, what it allows through, how much of it allows through, and it depends on the concentration of the solute that cannot cross the membrane. So those two factors are going to determine the flow of water, and what happens is going to be either a hypertonic condition, which means hyper is a lot. So when we have a lot of solute in the solution, it is hypertonic. If it's hypo, that means we don't have very much. It's very little solute in the solution. And if it's isotonic, it means we have equal amounts of solute in both solutions and we're at equilibrium. Okay, moving on. So again, this is just reiterating the same ideas. Direction of osmosis is determined by comparing the total solute concentrations on both sides of the membrane, and in our case, the cell membrane. So again, if you need review of these concepts, you can pause this and look over it, but basically it's something that should be reviewed. The next picture here shows you what happens when a cell is put into the different tonicities of solution. And the cell's survival is going to depend on making sure there's a balance of water, uptake, and loss. So in this picture, I want you guys to study this if you're not familiar with it. But when we put a cell into an hypotonic solution, that means there's not very much solute in our solution, then what happens is that the cell is going to take water in, because what is happening is there is a higher concentration of free water outside of the cell because there isn't very much solute outside of the cell. It's hypotonic in our solution. So you can think of fresh water as hypotonic because it doesn't have very much solute in it. It's just nice, clean water. But inside the cell, we have different solutes. So we have a lot of different molecules inside of a cell. And so that means the water is going to flow from where it's highly concentrated to where it's less concentrated because of the solute. It's going to move towards the solute. So where solute is concentrated is where water is going to flow. So in this case, in an animal cell, what's going to cause it to occur is a bursting of that cell. Because too much water flows in to the cell membrane, it gets filled with water and will burst. In a plant cell, we actually have a cell wall that prevents the lysis of a plant cell in a hypotonic environment. If it's isotonic, that means we have equal amounts of water flowing in and out of the cell. Very happy cell. That condition is called flaccid. When we're talking about a high amount of water pressure entering into the cell, we call that turgid. So the cell is actually being pushed out. And then when we put a cell into a hypertonic solution, that means we have a lot of solute in the solution, for example, with salt water, then the water is going to flow towards where there's more solute. So it's going to flow out into our hypertonic solution. So it's going to shrivel up our cell. In a plant cell, again, we have our cell wall here that's protecting it from actually losing its shape. But the cell membrane itself inside the cell, plant cell here, is actually going to pull away from the cell wall. And that condition is called plasmolysis. And so we say the plant cell becomes plasmolysed. So here are some questions for you, and actually the answers are there. But as you're starting to think about quizzes and tests, you need to know osmoregulation is the control of water's balance. So we are going to look at the protist, the paramecium, but basically a paramecium lives in a freshwater environment. So that means it has more solute within it than the environment it lives in. So that means water is going to flow into a paramecium on a constant basis. And so the paramecium is hypertonic. Its internal life, its internal cell, is hypertonic to the water that is outside. So why is that a problem? Okay, so if water is constantly flowing into the paramecium, what's going to happen to the paramecium? It's actually going to burst and die. And so it will be a huge problem for the paramecium if there's not some sort of way to deal with it. And so there actually is a way to deal with it. The parameciums have a specialized organelle that's called a contractile vacuole, and that's shown right here. And what this vacuole does is it takes all the excess water. That water actually, through osmosis, flows into this vacuole, and the vacuole contracts and squeezes the water and exports it, secretes it actually out of the paramecium. So it's really like a sump pump, if you've ever heard of one of those. It's pumping water constantly out of the paramecium. So that is what allows a paramecium to live in fresh water. Because it doesn't have a cell wall, it really has to use this contractile vacuole in order to survive. Okay, so again, osmoregulation is managing the balance of water. So in a hypotonic environment, when a cell's in fresh water, there's a high concentration of water around the cell. And the problem is that the cell is going to fill up with water because water is going to move from its high concentration to low. So again, osmosis is about diffusion, going from a high concentration to low. So we're going from high water concentration to low water concentration. And I'm going to go over this in class again because I think a lot of people get confused about what is actually highly concentrated or not. Because when we're talking about tonicity, we're talking about how concentrated the solute is. But when we're talking about the movement of water, we're talking about the concentration of water from high to low. So I'm sure that has just totally confused you. So again, we'll go over this in class and make sure that you understand it. But for plant cells, again, they have this cell wall. And this is when the cell wall becomes incredibly valuable to a plant because when the plant begins to fill with water, so it will keep filling up, and its plasma membrane is going to continue to expand and expand until it presses up against the cell wall. And that condition of pressing up against the cell wall is called a turgid condition, and it's actually the normal condition of a plant cell. So that's why we keep our plants watered, and if we don't, they wilt. And they wilt because the cells are actually not turgid. They're actually wilting, and we'll look at that when we look at hypertonic conditions. But basically, the turgid condition allows a plant to sit up straight and look healthy and perky. And it's because of this pressure on the cell wall being exerted by the water within the cell. So when we have the opposite condition, when we have hypertonic, I'll start with that plant cell again. So a hypertonic solution around a plant cell is going to cause water to flow out of the plant cell. So also in a condition when we don't water our plants, again, there's not enough water within that plant cell, so the same thing happens. So in this case, the pressure of the water is not keeping the cell membrane up against the cell wall. And so what's happening is that basically the cell is collapsing on itself inside. And that again is what gives us that wilted look of our plants that aren't getting water, which we see a ton of right now with the drought. That condition is called plasmolysis. It's that wilting, and it can recover from it simply by adding water to the inside of the cell. It's important to note when we're still talking about cell walls that, again, remember that bacteria and fungi have cell walls. So they also can plasmolyze in hypertonic environments. So they can also wilt or collapse on themselves. So now, looking at animal cells, what happens to them in a hypertonic solution? So again, hypertonic means we have a lot of solution out here. We have a lot of solute, not a lot of solution, a lot of solute outside of the cell, more concentrated solute than we have inside the cell. And so that means water is at a higher concentration inside than out. So it's going to flow down its concentration gradient to where it's a lower concentration. It's going to flow out of the cell, causing the cell to shrivel up. And this can happen, for example, with shellfish. If a shellfish is in too salty an environment, it actually will start to shrivel up. And so they actually have a pump as well within them that pumps salt out of the water that's brought in. Okay, and then our ideal situation is again isotonic. So this is when we have the flow of water going in and out of the cell through osmosis in equal basis. We have equilibrium. And when we have that condition, that means the concentration of solute is the same outside as it is inside. And again, another way to say equilibrium is to say our cells are in homeostasis. So we like this, for example, with our blood cells in the blood plasma. We like them to have an isotonic condition, which means the solutes in the blood plasma need to be the same concentration as the solute within the blood cell. So that's why our IV solution in the hospital will have a solute concentration to it. It's why we don't just pump water into somebody who's lost fluids. When we're pumping fluids into the blood, we actually have to add some solute to it. And in most cases, they're just going to put some salts. Actually, it's some different electrolytes, but primarily salt as the solute in order to maintain this isotonic condition between your blood cells and the solution being added. Okay, so I'm not going to spend any more time on aquaporins um, because we already talked about that. The only thing I'll mention is kind of interesting. It's only in 2003 that really we learned about this protein, the aquaporin, in a cell membrane. And most of the work was done, they're saying here with John Hopkins and the Rockefeller Center, but a lot of it was actually done at UC Berkeley as well. So again, there's so much in biology that you can get involved in discovering. There's so much to learn, and constantly there are new discoveries. So if that's something that interests you, then biology is the science for you. And our last 5 seconds here. I'm just going to leave you with this